If you live with chronic pain, if you have spent years navigating the impossible trade-off between relief and risk, then you already understand the cruel arithmetic of modern opioids. They work, and that is precisely the problem. Every opioid we have, morphine, oxycodone, fentanyl, buys pain relief at a price: addiction, tolerance, respiratory depression, the possibility that the very drug keeping you functional could one day stop your breathing. For 70 years, we have assumed that was simply the deal. The biology of pain relief and the biology of danger were locked together, inseparable. A paper published in Nature in April 2026 suggests that assumption may be wrong. The compound at the center of this story belongs to a chemical family called the nitazenes. And if you know that name, your first instinct is probably alarm. Rightfully so. Nitazenes were first synthesized in the 1950s. Chemists building on a flat ring scaffold, called benzimidazole, discovered they had created something extraordinarily, dangerously potent. The most powerful member of the family, etonidazine, proved to be roughly 1,000 times stronger than morphine. The entire class was immediately shelved. Decades later, illicit chemists rediscovered them. By 2020, nitazenes were appearing in the street drug supply, hidden in counterfeit pills mixed with other substances, killing people who never knew what they had taken. This is the family our compound comes from. And yet, the NIH team looked at this chemical family and asked a different question. Not, how do we stop these molecules? But what if we could redirect them? Their starting point was a compound called FNZ, fluornitrazine. It had one very useful property. Its fluorine atom could be replaced with a radioactive isotope, fluorine-18, and tracked through the brain in real time using a PET scanner. What they saw in the scan surprised them. FNZ entered the brain rapidly and then disappeared. Within 5 to 10 minutes, it had been cleared, and yet the pain relief it produced lasted for hours. Something else was doing the work. What the researchers had stumbled onto was a metabolite, a byproduct created when the body processes FNZ. Liver enzymes stripped a single molecular side chain from FNZ, breaking off one ethyl group. What remained was a new compound. They called it DFNZ. Every opioid you have ever heard of targets the same doorbell in your nervous system, the mu-opioid receptor, or MOR. This protein snakes back and forth across the neuron membrane 7 times. When an opioid plugs into it, it triggers a cascade of signals inside the cell. There are 2 main signaling pathways. The first, involving G proteins, is associated with pain relief. This is what we want. The second, involving a molecule called beta-arrestin, has been linked to the dangerous side effects: slowed breathing, tolerance, the scaffolding of addiction. For years, drug developers have tried to create biased agonists, molecules that favor the good pathway and avoid the bad one. Some progress has been made, but nothing had truly solved the problem. DFNZ does something different. In a laboratory assay, it activates the mu-opioid receptor more powerfully than morphine. It is what scientists call a superagonist. And superagonists are supposed to be the most dangerous opioids of all, except, in living animals, DFNZ behaves like something far gentler. The key to DFNZ's unexpected safety lies at the border of the brain itself. The blood-brain barrier is the brain security system, a tightly controlled membrane that decides what gets in. Embedded in that barrier is a protein called the P-glycoprotein transporter, or PGP. Its job is to pump certain molecules back out before they can accumulate. DFNZ turns out to be a PGP substrate. That means the blood-brain barrier is actively limiting how much DFNZ gets into the brain. It enters slowly, in small, steady amounts. The brainstem, the region that controls breathing, never gets flooded, and that changes everything. Fentanyl rushes in. It saturates the brainstem. Breathing stops. DFNZ trickles in. Enough reaches the pain processing centers to bring relief. The brainstem is spared. The researchers proved this with a striking experiment. They gave animals a drug that blocks PGP. When they did, DFNZ suddenly caused the same oxygen crash in the brain that fentanyl produces. The protection is built into the molecule's relationship with the barrier. Remove the barrier's gatekeeping, and the danger returns. Leave it intact, and you have something that looks, for the first time, like a safe opioid. Pain relief is only half the story. The other half is addiction. Addiction in the brain is largely a dopamine story. Deep inside your reward circuitry, in a region called the nucleus accumbens, addictive drugs trigger a sharp, rapid surge of dopamine. That surge is not just pleasure. It is a learning signal. a command to the brain. This matters. Remember this. Do it again. DFNZ still increases dopamine. It is still a powerful drug, but the kinetics are different. The rise is slow, the peak is moderate. The signal the brain receives is not a command, it is more like a murmur. In self-administration experiments, rats would press a lever to receive DFNZ, but the moment it was replaced with saline, they stopped. Almost immediately, they did not keep pressing. There was no extinction battle, no withdrawal spiral. Repeated dosing produced tolerance only minimally. 14 classic withdrawal signs were tested. Only one appeared: mild irritability. That is not a tweak. That is a fundamentally different pharmacological story. The NIH researchers are direct about who could benefit most if DFNZ makes it to human trials: surgical patients, people with cancer pain, and chronic pain patients. The team is now preparing an investigational new drug application, the first formal step toward human clinical trials. That process takes time, years, most likely, and there are real questions still unanswered. Everything described in this paper was done in rodents. Human biology is more complex. CYP enzymes vary between individuals. For a long time, the story of opioids has been a story of compromise. You could have the pain relief if you accepted the risk. That was the deal. DFNZ is the first serious scientific challenge to that assumption in a generation, not because it is weaker, but because it engages the receptor in a way no one expected. The science is real, the road to your pharmacy is long, but for the first time in decades, the road exists. If you are living with pain right now, keep watching this space. This one is worth following. This discovery was originally published in Nature in April 2026 by Gomez and colleagues. Please remember that this video is for informational purposes only and does not constitute medical advice. Always consult your physician. Visit unbedsidemanners.com for more stories and to learn about physicians railroaded by bad policy.